STAR stands for Scientific Training in Antimicrobial Research Strategies, and its main purpose is to provide scientific training to 9 PhD fellows and 1 postdoctoral scientist in antimicrobial discovery and development. The project has 2 main sub-programs, one on target-based approach to developing new antibacterial drugs and one on anti-malarial drugs. Both themes are addressing the problem of drug resistance. Drug resistance in bacterial pathogens continues to make headline news, and even our last resort drugs for multi-resistant pathogens seem increasingly fallible. The malaria parasite infects more than 200 million persons every year and causes over 1 million deaths annually. Malaria drug resistance is spreading now across the globe, and it's linked to the increased incidence of morbidity and mortality. Here we present our target-based discovery and development of new antibacterial drugs.
There's an urgent need for development of antimicrobials and anti-infectives to readdress the balance between drug resistance and the available therapeutic options. Our group focuses on novel antimicrobials to combat antibiotic-resistant bacteria.
Antibiotic resistance is the term given to pathogenic bacteria that are no longer inhibited by currently available antibiotics. Eventually, bacteria can become resistant to antibiotics, particularly when used extensively, for example in animals for food production. If a prescribed course of antibiotics is not completed, then it can also facilitate resistance development. During the second half of the 20th century, antibiotics were discovered by screening naturally occurring compounds. Although this was fast, resistance to these natural antibiotics rapidly emerged and spread among bacteria.
Novel approaches to discover effective antimicrobial drugs are applied to control the diseases caused by resident bacteria. Target-based drug design is more specific and is aimed at essential elements of the bacterium, such as 2-component systems. The 2-component systems are important for gene regulation in the bacteria. The bacteria need them to perceive and to respond to the changes in environmental conditions. These 2-component systems are not found in mammals. Drugs aimed at 2-component systems are highly specific and less likely to induce resistance in bacteria.
We focus our research on 3 extremely important pathogens: Mycobacterium tuberculosis, Streptococcus suis, and Streptococcus pneumoniae. Mycobacterium tuberculosis causes tuberculosis or TB, a lethal infectious disease that leads to more than 2 million deaths per year worldwide. Current treatment is directed against actively replicating TB bacteria, but there are no drugs against the dormant bacteria in the latent stage of TB. Another important pathogen, Streptococcus suis, causes serious problems for both humans and pigs worldwide. Increasing frequency of infections with resistant S. suis poses medical and societal challenges. Streptococcus pneumoniae is the first cause of community-acquired pneumonia, worldwide mortality due to invasive infection, and meningitis in children.
Structure-based drug design starts with the identification of an essential molecular target in bacteria, such as 2-component systems. Potential inhibitors of these targets are identified by screening large number of compounds. Target proteins are cloned in laboratory bacteria, overexpressed and purified to obtain large quantities of protein. Crystals of these proteins are produced in the presence of potential inhibitors. X-ray crystallography is a method to study the 3-dimensional atomic structure of the protein inhibitor complex. We can use this information to understand the interaction between the inhibitor and the target and to optimize the design of more potent inhibitors.
Another promising antimicrobial strategy is based on naturally occurring antimicrobial peptides. They are small molecules of the innate immune system, which plays a major role as the first barrier against invading pathogens. The mechanisms by which these peptides kill bacteria can be the basis for the development of a new kind of antimicrobials.
To date, we have cloned and expressed several 2-component systems from important pathogens, and for some of these we have obtained crystals. Structures of proteins from two-component systems are being used for virtual screening and potential inhibitors have been identified. We are discovering more about the roles of these two-component systems in pathogens.
Additionally, the mechanisms of bacterial resistance to host antibacterial peptides are being unraveled and might lead to new strategies to combat bacterial infectious diseases.